I am going to spend the next 3 lectures talking to you about immunometabolism and how metabolism is important for immune cell function. So you'll probably all have at some point learned about basic metabolism. I think it happens during the second year of your degree. But in this first lecture, we're going to sort of recap the core principles of cellular metabolism and explore how it is used in different immune cell subsets. In my second lecture, we're going to focus on lymphocyte immunometabolism, and then in my third lecture, we're going to start thinking about how the implications that this has for lymphocytes and immune cells working in immune microenvironments such as the tumor microenvironment and inflammatory microenvironments. You're also going to, as part of this sort of series on immunometabolism, get some lectures from Luke and Neil on macrophage immune metabolism and targeting metabolism to treat inflammatory and infectious diseases. Okay, so let's start off with a basic slide about cellular metabolism. So this here is our mitochondria, my crude drawing of a mitochondria. And there are lots of fuels that cells can use. So obviously the one that comes to mind first is glucose. Glucose is a very important fuel for cells that's metabolized by glycolysis. And the product of glucose, which is pyruvate, can have a couple of fates. So it can either be converted into lactate and secreted from the cell, and this is often termed aerobic glycolysis, and we'll go into that a bit more in a minute. Or else, in other cells, it can be closely coupled to this process in the mitochondria called oxphos, where the product of glycolysis pyruvate is shuttled into the mitochondria, converted to acetyl-CoA, and then drives this process of oxphos, which is very good at making energy. So glycolysis itself makes some energy, but oxphos is very efficient at making energy. So if you're a cell that wants to make lots of energy, you will usually couple glucose glycolysis to the TCA cycle and oxphos, which is an efficient way of making energy. It's not all about glucose, so there are other fuels that the cell can use to generate energy. Amino acids can feed into the TCA cycle here to fuel oxphos. Fatty acids can be broken down in the mitochondria to give you acetyl-CoA as well. Glutamine can also feed into the TCA cycle as alpha-ketoglutarate. And there are also some amino acids that can feed into glycolysis. So there are a range of fuels that can be used by the cell to generate energy. But it's not all about energy. Of course, cells need to do more than just have the energy to go about their business. Especially immune cells. Immune cells can engage in a really robust biosynthetic response when they become activated, because they need to make lots of effector molecules like cytokines. Often they would also need to undergo robust proliferation. And when one cell becomes two, obviously you need to replicate all the proteins, the DNA, the nucleotides, and also the lipid bilayers. So often an activated lymphocyte or an activated immune cell, there's a huge biosynthetic burden. And these basic fuels like glucose and glutamine can feed into the cell, and their metabolites can then be siphoned off from these core metabolic pathways that drive energy production to be used for biosynthetic purposes. So here is the mitochondria. The mitochondria is a very interesting organelle. It's got this clear double membrane structure. As you can see here, you've got your outer membrane and your inner membrane. And this is really essential for the process of oxphos and the process of ATP production in the mitochondria. And we'll show that now in the next few slides. So this is looking a bit more closely at glycolysis. As you can see, glycolysis is a series of enzymatic steps that converts glucose into pyruvate. You also have a net production of 2 ATP. It also involves cofactors like this enzyme here, glyceraldehyde dehydrogenase, GAPDH, which we'll hear a bit about in subsequent lectures. It's an important enzyme in glycolysis, and it also requires this cofactor NAD+, which converts to NADH as part of its enzymatic process, and that becomes important in a minute too. So this makes some energy, but then you couple the pyruvate to the TCA cycle, also called the citric acid cycle, and your pyruvate is converted into acetyl-CoA, producing CO2, and then there's a series of enzymatic steps that goes around the cycle where acetyl-CoA is feeding in 2 carbons, and you have all these steps, all generating these reducing equivalents NADH and FADH2, which are able to carry electrons and donate those electrons into the electron transport chain here, which is the complexes 1 to 4. And these are embedded in the inner membrane of the mitochondria. And as these electrons flow through this electron transport chain, they also pump protons across this membrane, generating this gradient. So you have lots of protons in the intermembrane space here, and lower levels in the matrix here, which is the center of the mitochondria. And that's the core driving force that drives ATP synthesis through this multi-subunit enzyme here, which is ATP synthase. So your fuels come in through the TCA cycle, generate the reducing equivalents, which bring the electrons to the electron transport chain, which pumps across the protons, and the protons then come back across the membrane through this complex here to generate your ATP. As I said before, pyruvate has another phase as well, and we'll discuss when and why this is used. Pyruvate can also be converted into lactate, and lactate can be secreted from the cell, and that is also associated with the conversion of NADH into NAD+. So the cofactor for pyruvate conversion to lactate is NADH. So let's just focus on this part here for a second, the electron transport chain and ATP synthase. So here's a nice video you can get of one of these GIF websites, trying to illustrate the dynamics of all these molecules occurring during this process of OXPHOS and ATP synthesis. So here you can see this is the matrix on this side, and here is the intermembrane space. And as you can see, complex 1, 3, and 4 are all proton pumping complexes. So as they receive the electrons from these reducing equivalents that you can kind of see coming in here, they are also pumping protons across here. Protons are the yellow dots here. So you can see that you have an intermembrane space that's a high concentration of protons and a low concentration in the matrix. So you have this clear proton gradient. And then you can see here through this ATP synthase, which is this complex multimeric machine, as it were, these protons are pushing through the ATP synthase, causing this rotation to occur and these conformational changes that allows ADP to bind, inorganic phosphate to bind, and as it rotates, these are combined to give you ATP. All driven by protons passing this way from the intermembrane space into the mitochondrial matrix. So it's essentially quite simple. You've got a series of complexes here that pump protons across, and this is used as the force to make your energy, the proton motive force that drives ADP to combine with inorganic phosphate to give you ATP. And here's another representation of you can see how this rotation is causing these complexes to move, revealing binding sites and providing the energy to convert ATP to ATP. And here you can see even more detail. You have your ATP and your phosphate binding, and then this change in conformation and releasing the far side of your ATP. It's really a really elegant machine. That really explains how our mitochondria are so efficient at generating ADP. Now, contrary to what you might see in some of the textbooks, our mitochondria are actually very dynamic and very interesting organelles. They're not just sort of oval-shaped organelles in the body. They can be, certainly, but they can also form these elongated sort of networks of mitochondria. So here's all the mitochondria here on the left. You can see in this case, in this cell, they are forming these elongated mitochondria with complicated networks. So you can see some branching and forming this really sort of elaborate network here. Again, here you can see also, this is a time-lapse video. You can see how mobile they are. They can move around the cell. You can also see that at some points you can see fusion, where two mitochondria join together. There's an example up there. You can also see fission, where you can see some mitochondria breaking off. Here we have a small sort of mitochondria up on its own. So it's a really dynamic process where they form these networks. They can break apart, they can fuse together. And you also see even beyond that, you have here at the bottom, you have a closer look at at the internal structure of these mitochondria, and you can see that they have quite interesting internal structure. You can see here they go from, in the course of this video, from these elongated structures with these cristae here, which are the invaginations of the inner membrane, forming these sort of close-knit mitochondrial cristae, as you can see here. And then over time these mitochondria change in structure, and you see a loss of this internal structure and these close packed mitochondrial cristae. And that would have implications for the efficiency of OXPHOS. And in some of the subsequent lectures, we're going to be looking at this a bit more closely, because it seems to be that in certain pathological situations, lymphocyte dysfunction is associated with change of the morphology of mitochondria, and in particular in the internal morphology. So their organized structure is lost, and this affects their efficiency of OXPHOS. But you can imagine, if you have these really tight mitochondrial cristae, you have all your complexes highly ordered together. Electron flow is efficient. The flow of protons across the membrane is efficient. But as you lose this internal structure, the greater distances occur between these the complexes, and the electron flow is less. This results often in things like reactive oxygen species being formed instead of the efficient process of OXPHOS. But we'll talk about that later. Take home message from this slide is mitochondria are very dynamic and complicated structures. So as I said before, mitochondria can undergo this process of fission and fusion, and that's how they form those sort of beautiful networks that you saw in the previous slide. So you can have two mitochondria joined together to give a longer mitochondria, and ultimately leading towards these kind of elongated structures here. And there's lots of different machinery that are required for this. You can also have fission, where you have large mitochondria breaking off into smaller mitochondria, and this is an important process in maintaining mitochondrial health, because often if you have a damaged part of the mitochondria, it'll be pinched off through a process of fission, and then these damaged mitochondria can be degraded by a process called mitophagy, which is basically the way your cell can clear up these damaged mitochondria and repurpose the individual components. So this whole process of fission and fusion is important for driving efficient OXPHOS with these elongated networks of mitochondria, and also make sure that you can maintain mitochondrial fitness through degrading damaged components of the mitochondria through mitophagy. So what is ATP? So ATP is basically your core energy store. And the reason it's an energy store is because this final terminal phosphate bond is a high energy bond and contains lots of energy within it. So if you break open this bond, you can release energy. Kind of, the analogy I often give is that of glow sticks. So glow sticks, if you look at them, they're very rather uninteresting until you crack them, and then all the sort of the chemical energy that's stored within it is released in the form of light. In the case of ATP, you have this energy being stored in this terminal phosphate bond, and once that's broken by enzymes, it can be used to do work. It can be used to pump ions across the membrane in the context of ATP coupled transporters. It can be used to drive enzymes like kinases and other things like that. So it's kind of like your core unit of energy within the cell. So if you are a cell and you really have a high demand for ATP, and that's what you want to generate, this is a way that, following this red line here, is a way that you could efficiently make ATP. So if you metabolize your glucose to pyruvate and you couple that to the TCA cycle, all the way through oxphos, you can make up to 36 ATPs per glucose molecule. So very, very, very efficient. And obviously, the key point here is this requires oxygen. So, the terminal electron acceptor in this electron transport chain is oxygen, which is then converted into water. So this is the main reason why we require our cells require oxygen, is for this process of oxphos. This is also, of course, the main reason why we make carbon dioxide. You can see here that this TCA cycle is the main source of CO2 in our cells. So what happens in the absence of oxygen? So there are places in our body where it can become hypoxic, in the gut, in pathological situations like within tumors, you can become hypoxic. There's low levels of oxygen, in which case, in the absence of oxygen, oxphos can't continue. So the response the cell does to this is to engage anaerobic glycolysis. This is where they ramp up glycolysis and convert pyruvate into lactate, because it can't obviously enter into the mitochondria, because this process can't continue in absence of oxygen. Now, this is an inefficient way of making ATP, but if you ramp up the flux to this pathway, you can still maintain energy homeostasis. So this is termed anaerobic glycolysis, and you generate lots of lactate, which is secreted within the cell. And that was originally thought to be the two different sort of types of metabolism. You could have aerobic conditions and anaerobic conditions where you just do glycolysis, aerobic conditions where you do glycolysis coupled to oxphos. It became clear a long time ago by work by Otto Warburg that tumors were a bit different, and tumors seemed to be making lots of lactate despite the fact that there was plenty of oxygen, and they termed this aerobic glycolysis. So converting the majority of glucose to lactate despite the fact that there's plenty of oxygen. That's why it's called aerobic as opposed to anaerobic glycolysis. And it turns out that what's the benefit of this? So this is basically what it is. So in aerobic glycolysis, even though there's plenty of oxygen around, you are driving glycolysis very fast, and the majority of the pyruvate that's made is converted to lactate, and a minority of the pyruvate might be entering into the mitochondria and engaging in oxphos. But in fact, a couple of years after that first paper where the Warburg effect was described, this effect of tumor cells, Otto Warburg also showed similar things in activated leukocytes, which wasn't really picked up on so much at the time, because there's a huge focus on cancer research back in then. We're going back to the 1950s. And so it turns out actually, this is a signature of highly proliferative tissue or cells that are engaged in a lot of biosynthesis, that they engage this aerobic glycolysis, because this is a benefit to cells in terms of biosynthesis. So it's not just tumors, it's proliferative tissue, which of course is very relevant for activated immune cells. So on cells that have a high biosynthetic demand will engage in this aerobic glycolysis, where they take up lots of glucose. A lot of it, or the majority of it, is converted to lactate, and some of it then goes on into the mitochondria to efficiently make some ATP. So aerobic glycolysis supports cellular biosynthesis. So you're going to see that that's a common theme within our immune cells. When we talk about immune cells, the subsets that are engaging in lots of biosynthesis in general will have ramped up aerobic glycolysis, will be using lots of glucose, and may also producing lactate. So we got to think about this. So the core metabolism of our cells is to make energy. So we can think about glucose as a fuel source for making energy. We also got to consider that glucose can also be a source of material for biosynthesis. So we can use the carbons of glucose to make amino acids like alanine, like serine, ultimately other amino acids like aspartate. You can use it to make you can convert the carbons and glucose into nucleotides, and also can be used to generate lipids. So glucose can also and also amino acids like glutamine can be sources of carbon for building the components of the cell. And that's very important if you are a proliferating cell, where you need to build all these components for one cell to become two cells. So if we redraw glycolysis and the TCA cycle slightly different to focus on the carbons, we can see that glucose is a 6-carbon sugar that is excuse me, is metabolized to pyruvate, which is a 3-carbon. Then here we have the TCA cycle, and we think about the different points in this pathway where these carbons can be withdrawn from these pathways and used for biosynthesis. So after the first step, where we have glucose 6-phosphate, this can enter into the pentose phosphate pathway, which is a pathway that's important for generating ribulose 5-phosphate, which is one of the core components that's required for making nucleotides. So DNA, RNA, very important for proliferating cells, for instance. We can also withdraw carbons at this point to generate glycerol, which is the backbone for phospholipids. Down here we can start making amino acids like serine. Pyruvate can be converted to alanine, which are important just for making protein, but serine is also important for making phospholipids, for making phosphatidylserine. And also from the TCA cycle, citrate is an important molecule that can be withdrawn from the mitochondria and used for building a range of things, including cholesterol and fatty acids. So it's broken down in the cytosol to acetyl-CoA, which is the core building block for making fatty acids and cholesterol. This also generates oxaloacetate, which can be converted into aspartate, which is obviously an amino acid, but it's also important for making nucleotides. So we can start to see the picture from this slide is slightly different to the previous ones, because we're focusing on how these fuels can be used for biosynthesis as opposed for making energy. So if I start to tell you that glucose is important in these cells as a source of carbon for biosynthesis, well then it starts to, if you think about 80% of the carbon then being secreted in the form of lactate, you start thinking, well, this seems like a bit of a waste. You're secreting some of this carbon that can be used for biosynthesis. So why do cells do this? In fact, in some cells, this is some of the metabolomics we did in our NK cells, and you can see here, when we activated our NK cells, almost 80% of all the glucose molecules that were taken up into the cell were secreted as lactate. So why do they do this? And there's 2 reasons for this. The first reason is that the end product of glycolysis is pyruvate. But the problem is that glycolytic flux can go much faster than pyruvate can be metabolized within the mitochondria. So if you're trying to ramp up glycolysis to use the carbons of glucose for biosynthesis, pretty soon you come to a limiting point where you have too much pyruvate. It can't be dealt with by mitochondrial metabolism. Therefore, you get product inhibition of the whole pathway. So a buildup of pyruvate will start to inhibit all these steps, and pretty soon your glycolytic rate falls. So one way to sort of deal with that is, well, you can get rid of that pyruvate by converting to lactate. So you're getting rid of that product inhibition, allowing for high rates of flux to continue. Another reason why you convert pyruvate to lactate is because it converts NADH to NAD+. So this is a cofactor that's sort of interconverted between NAD+ and NADH. And the problem is, if you have high glycolytic flux, at this point here, which we mentioned previously, GAPDH, you get a conversion of NAD+ to NADH. And if you have high flux to glycolysis, you end up having all your NAD cofactor in this state, NADH state. You now have no longer any NAD+, and GAPDH can no longer exert its enzymatic activity, and you get inhibition here. So the second thing, the beneficial thing that happens by converting pyruvate to lactate is that you regenerate NAD+. So I guess the key point here is by converting pyruvate to lactate and secretion lactate, you can maintain high rate of flux through glycolysis. And high flux means all the intermediates within glycolysis tend to be at elevated levels, allowing those pools of intermediate to be siphoned off into other pathways for biosynthesis. So it's not necessarily the most efficient way of converting carbon into biomass, but of course, remember, especially in immune cells, it's not about efficiency; it's about speed. If you have a T cell response, T cells are activated because you have an infection, you have an immunological challenge, and if it's an infection with bacteria which divide very, very quickly, you need your T cell response to ramp up very quickly. So it's about how quickly you can build 2 cells from 1 cell, rather than how efficient you can do it. So secretion of lactate may seem wasteful, but it's a way of maintaining high rate of flux to glycolysis. Because you're taking in lots of carbon, you can also have high concentrations of intermediates that can be siphoned into these biosynthetic pathways. So we step back and look at the sort of the core metabolic pathways here and think about what's the purpose in immune cells. Well, of course, the first thing which we discussed was that they're important for energy production, ATP production. And we've just discussed now how all these different intermediates can be also converted into core molecules required as building blocks within the cell, so important for biosynthesis. There are other reasons as well. So obviously, there are other fuels as well, not just glucose. We have amino acids, we have glutamine that can feed into different amino acids, feeding into different parts of these pathways, which can be used then to make all these different biosynthetic molecules. Also, something which we're going to touch on in the third lecture is that some of these metabolites and metabolic enzymes have more direct roles in controlling immune function, because the metabolites can actually alter the function of proteins or enzymes, alter the expression of certain metabolic genes. The enzymes can also have direct immunological roles outside of the role in these metabolic pathways. So actually, and we'll go into this a bit, but it's an ever-emerging field. There's lots of examples of this. So actually, metabolism can directly regulate immune function through the production of these metabolites, which can act to regulate expression of genes, activity of proteins, and also some of the enzymes, as I said, can have roles outside of their metabolic role and can directly control the immune functions of these cells. So we have metabolism controlling energy, biosynthesis, and controlling the functionality of the cell. And another thing that metabolism is important for is reactive oxygen species, redox signaling, redox balance, maintaining redox control. So obviously, reactive oxygen species are important for signaling molecules, but also they can cause damage. Particularly, they can cause damage to the mitochondria. So you also want to be able to balance the production of ROS with efficient quenching of that ROS to prevent excess damage. And metabolism can be important. These core metabolic pathways can be important for that, because they can allow you to maintain your pool of glutathione, and you're reducing agents within the cell that can quench this reactive oxygen species. So this is the pentose phosphate pathway that I discussed previously. It's quite an interesting pathway that has quite a few outputs. So glucose-6-phosphate can be converted into ribulose 5-phosphate, as I said before, to make nucleotides. During this process, you also generate NADPH, so it's similar to NADH, but different. It's got extra phosphate, and this is an important cofactor for biosynthesis, and it's also, in particular, fatty acid synthesis, but also other biosynthetic pathways. It's also an important cofactor for maintaining a pool of glutathione, which is important in its antioxidant capacity. So this is why, because the glutathione reductase, which reduces glutathione to maintain its ability to deal with reactive oxygen species, requires NADPH as a cofactor. So the pentose phosphate pathway can be used in multiple formats. So so this shows the whole pathway and the different routes that the carbons can go. If you have a core requirement for nucleotides, you can convert your glucose-6-phosphate into ribulose 5-phosphate, and this can be siphoned off to make the nucleotides, making NADPH at the same time. Or if you don't have a requirement for nucleotides, and maybe you have a core requirement for NADPH, this ribulose 5-phosphate can be recycled back into the glycolytic pathway through this arm of the pathway here. So you can have glucose-6-phosphate going through the pentose phosphate pathway and back into lower down in glycolysis, and ultimately ending up in pyruvate and lactate. And this allows you to generate NADPH. Alternatively, if you don't have a requirement for NADPH, you can also siphon off intermediates lower down in the glycolytic pathway just to make nucleotides. So depending on the actual demands for the cell has, you can make the molecules that you need. So it's quite cellular metabolism is quite versatile in that way. It will meet the demands of the cell. So I think hopefully we got a good introduction into metabolism and how metabolism can be tuned to provide what the cell needs in terms of energy, in terms of biosynthesis, in terms of dealing with reactive oxygen species, and also, which we'll learn about later, a bit about how these metabolites can also regulate the cell function. But we have loads of different types of immune cells, ranging from your lymphocytes, the naive lymphocytes, to your activated lymphocytes, which are so different. They go from being very quiescent to very active cells. You've got your innate cells, which are already ready for action, like macrophages and neutrophils, which are your first responding innate cell. You got your NK cells, range of immune cells, and you will now be learning about all the different functions of these immune cells. So it's not surprising, I suppose, that the metabolic signature of each of these immune cells tends to be quite different because they have different functions, whether that be different levels of biosynthetic demand, different requirements for energy, or the fact that they operate in different environments where different levels of nutrients or even oxygen might be available. So in the next few slides, I'll kind of sort of describe in very simple terms some of the signatures that we know about immune cells and how they can differ. Even just taking the one lymphocyte population, the T cells, we can see that T cells, depending on whether they're naive, memory, effectory, regulatory cells, have very different metabolic signatures. So a naive T cell is a very quiescent cell. They rely on glucose, not shown here, but they can also take up some fatty acids, and they efficiently convert these fuels using oxphos to make energy. They also use some glutamine, but they have relatively low level of nutrient uptake. They don't express high levels of transporters for these nutrients, but they efficiently use it to make ATP because these cells aren't doing much, and most of the demands is for energy. You engage in if you activate your T cell through TCR and appropriate co-stimulation, you go from being a naive cell to a cell that engages in rapid and robust proliferation. Clonal expansion. This is important because it takes a particular T cell, which is a particular T cell receptor, which is at low numbers, and it ramps up those numbers to give you an army of those T cells with specific ability to recognize the pathogen that you're infected with. So clonal expansion is crucial, and has a high biosynthetic demand, and that's why they robustly induce the uptake of nutrients like glucose, glutamine, also a range of other amino acids. So in general, all the nutrient transporters are increased in expression. They engage in aerobic glycolysis and they make lots of lactate, and this is a signature that's sort of tailored towards biosynthesis to allow them to generate the lipids, the nucleotides, and the proteins that are required for clonal expansion. If you move down towards memory cells, so memory cells are sort of what comes out the far end of an immune reaction. These are cells that have been selected by a particular immune challenge, and they have a TCR that's recognized the pathogen, and you then generate these memory cells so that should you ever become infected with that pathogen again, you have this army of memory cells that are primed and ready to respond even more quickly, and often preventing you getting symptoms of that infection the second time. And they have, depending on what study you look at, they seem to be doing every single pathway that you can think of. From they have glycogen stores which they can burn glycogen, they can make glycogen, they can make triglycerides, they can take up fatty acids. So really quite a complicated signature where they seem to have the machinery for lots of pathways already upregulated and ready to go. And I'll discuss this a bit more in a second. And then you got your regular T cells, which are different again. These are important in the control of the immune response, making sure it doesn't get out of control. And these cells have, compared to your effector cells, have lower levels of glucose utilization, and in particular they use fatty acids as important fuel. So they transport fatty acids, scavenge fatty acids from the environment and use that as a fuel. So you can see different T cell subsets can have quite different metabolic signatures. You then start to bring in some of the other innate cells, and you can see even more interesting metabolic signatures. We worked a lot on these natural killer cells, and found that they don't really use the TCA cycle much, but they do have high levels of oxphos, and they do a separate shuttle called the citric malate shuttle, which I'll tell you a bit about, because that's some work we've done in the lab in lecture 2. Luke's going to tell you a bit about the macrophage metabolism, and they kind of, depending on the type of macrophage you have, they can shut down the TCA cycle altogether, and that allows them to make metabolites that are really important in regulating the function of the macrophage. You can make succinate, you can make citrate for making prostaglandins, you can make itaconate, all these biologically important metabolites that can regulate the function of the macrophages. Whereas if you look at sort of the M2 types of tissue regulatory macrophages, they are different. They maintain TCA cycle and do beta oxidation. So very different signatures, because obviously your inflammatory macrophages have very different. Your inflammatory M1 macrophages have different functions to your less inflammatory, more regulatory M2 macrophages. And then you things, of course, remember we mentioned before, your neutrophils, which are your first line of defense in infection with bacteria, for instance. And actually they're interesting because they're packed full of lots of glycogen, so they have huge glycogen stores, so they are less reliant on external glucose, and they can actually drive glycolysis just by the glycogen stores that they have within the cell. And here's a really nice electron micrograph from this paper going back to 1983, I think it was, showing that neutrophils are packed full of glycogen. So all these black dots here, that's all glycogen stores. So you can imagine when these neutrophils arrive at a site of infection, if you have a bacterial infection, you can have a huge amount of bacteria localized within that site of infection. Bacteria use oxygen too. Bacteria some bacteria use glycolysis as their core energy metabolic pathway, so they have a requirement for glucose. So in that site with all these bacteria, you can imagine glucose is going to be rather in short supply. Oxygen might be in short supply as well. So in those circumstances, neutrophils are ready for this, because they can use their internal glycogen stores to drive aerobic glycolysis. So they're less reliant on external glucose, which may be low. They're less reliant on oxygen because they don't necessarily need to do aerobic glycolysis. So this seems to make sense in supporting the unique functions, the unique environments that neutrophils might find themselves in. So coming back to these memory T cells, with these complicated configuration, how do we make sense of this? As I said, some of these pathways are actually counteracting each other. You have glycolysis, converting glucose to pyruvate, and gluconeogenesis here happening at the same time, which is converting pyruvate into these intermediates at the top of glycolysis. And these have all been described in different research studies. So what I like to think is that they aren't necessarily going on at the same time, but the memory T cells have all these pathways there ready to go, and that affords them a level of flexibility. And of course, remember, these are cells that have been through an infection, so these are very important cells in your body. These are one of the core components of your immunological memory that's going to protect you from future infections. So very important cell. You would like them to be flexible in their metabolism so that they can live for a long time. You want the memory cells that you generated when you were a teenager, when you're infected with whatever virus you had, to still protect you in your in your 60s and 70s. So you want the longevity is very important for immune cells. So when I try to think of an analogy here, I think of these hybrid cars. This is the Prius. Actually, I recently bought a RAV4 hybrid car, and I really wanted to, there's wonderful displays on the front where it shows you where the energy is coming from, where the fuel, where the power is coming from the car, and the really nice graphic in front of the steering wheel. I wanted to take a video of it, but then I decided that driving a car while videoing with your iPhone is probably not a preferable, a safe way to generate your slides for a talk. So I've gone back to some of these older sort of graphics that you can get off the internet. This is, when the first Toyota Priuses that came out, which were the first hybrid models, they had this this lovely graphic on the dashboard. And I remember you used to often be getting taxis home, and all the taxis were Priuses, and you'd see this, and this tells you, here's the engine, here's the battery, and here's your sort of your generator for generating the electricity, and you can see very clearly what's driving the wheels at any particular point. So when you're going fast, you have your petrol engine is driving the car. Or other situations, you may be charging a battery from using the engine, and then other times you may be both driving the go forward motion of the car using the petrol motor and the battery motor. So really, and you can see this was in constantly in flux. As you accelerated, as you braked, you started to charge the battery. As you took off again, you'd be driving the wheels, and you'd be depleting the battery. And so I kind of thought this is probably what you have in the case of this memory cell. So they have this metabolic flexibility. So when they find themselves in environments where they have plenty of fuel, they probably use that fuel and that excess fuel, like glucose, to generate glycogen, to top up their internal stores, so that if they ever find themselves in an environment that's sort of depleted of nutrients, they have plenty of flexibility to deal with that, because they have all these internal stores, either in form of glycogen or triglycerides. So when for instance, if they've lots of glutamine available, that's a fuel that can be used to make energy, but also used to top up the stores of triglyceride. Glutamine can also be used to top up your stores of glycogen through gluconeogenesis. So they have this ultimate flexibility to be able to take whatever fuels they have, use them to make energy, and any excess they can then use them to top up their fuel stores. So obviously this is, I've no hard evidence of this, but this is my interpretation of all these papers that have showed all these complicated pathways to exist within the T cells, and these memory T cells. And I think, to me, it makes quite a nice bit of sense. Okay, so then focusing on another two cells that are again look a little bit different. So your NK cells, as I said, we'll talk about this in a bit more detail. The activated are, when we cytokine activate our NK cells, they ramp up glycolysis, take up a lot of glucose. Most of it's converted to lactate, as I showed, but they also maintain high levels of oxphos. So they're still using lots of oxygen. But interestingly, what we found here is that they weren't really engaged in the TCA cycle, but actually they had this other cycle called the citric myeloid shuttle that actually sort of operates across the membrane of the mitochondria. So it's half of it's within the mitochondria, and then in the cytosol as well. And this is able to drive oxphos, but at the same point generates cytosolic acetyl CoA. So and the exact reasons for this is still not clear, but we can start thinking about those now that we've sort of described the different kind of metabolic signature. And as I said, another very interesting and different metabolic signature is that of an activated M1 macrophage, where they have these two breaks in the TCA cycle. So this TCA cycle is no longer a cycle; it's converted into linear pathways. That's really important for generating particular metabolites like succinate, citrate, and itaconate. And you'll hear more about that from glucose. Sorry, you'll hear more about that from Luke in his lectures. Okay, so hopefully in this lecture we have explored the core principles of cellular metabolism and how they pertain to immune cells, which have such wide-ranging and different functions. How metabolism can be tuned for making energy efficiently, should that be what the cell needs, or biosynthesis, or for making effector molecules like in the macrophages. So we can see that immune metabolism is particularly interesting to look at, because cells undergo such dynamic changes in function during immune activation that corresponding to that you've got very clear and interesting changes in cellular metabolism that accompany that. So, when studying all the immune cells, we've really come across some really interesting and sometimes quite surprising metabolic phenotypes that seem to be there just to support those unique functions of the cell or the unique environments that those cells need to operate in. And that's something that we'll discuss, the sort of the immune or nutrient microenvironment that the cells operate in a bit more in lecture 3. So at the end of each lecture, I'll give you some suggested reading. Some of these are quite dense review articles. You wouldn't want to get lost in them, but if you are interested in, for instance, the exact reason why cells engage in Warburg metabolism or aerobic glycolysis, this is a really great basic review. Going back some time now, but it really explains why a cell engages in this type of metabolism, what's the benefit for it. So it really help to give you clarity on that. And then there's a range of other immunometabolism references. This is one we wrote early on. It's quite good for the basics, so it doesn't go into too much detail, but it covers a lot of the sort of the core principles of metabolism that we discussed in this lecture. And then so here's a good one from Luke on sort of a guide to immune metabolism for immunologists. So a little bit of extra reading which you can dip in and out of should you have any further questions, or should you find a certain area that you're particularly interested in exploring a bit more. Okay, well, thanks for your attention, and I'll see you again for lecture 2 in this immunometabolism series.